Electric Avenue, and we're going to rock on 2 Electric Avenue, and then we'll take it higher, as they say. Okay, right, I think we've got this fixed. Everybody should be able to see this. So I wanted to talk in general about host-parasite relationships, and then we'll move on to start covering specific groups of protists next week. You can think of parasites as being adapted to living on island paradises. If you think about it, if you're a well-adapted parasite, the host is doing everything for you. The host is providing you with food, and the host is also engaging in homeostasis, right? That physiological balance, maintaining a stable internal environment. So if you're a tapeworm living in someone's intestine, you don't really need a mechanism to maintain a particular temperature because your host is doing that for you. You don't need a mechanism to maintain a particular pH because your host is already doing that. So basically, from the point of view of a parasite, this is what you look like. Okay? You are in a perfectly comfortable habitat. All of your requirements are taken care of, right? You have, your host is doing all the work of bringing you food, and all you have to do is reproduce as much as possible. And it's not shown in this picture, but I'm guessing that that couple is going to be attempting to reproduce within about 5 minutes after the camera shuts off. Okay, so yeah, this is what you look like from the point of view of a parasite. Everything is done for you. The result of this is that parasites tend to lose structures and abilities that free-living organisms need. If there's no particular advantage to having a feature, it's not going to be maintained by natural selection, and it may be selected against. Right? If it costs a lot of metabolic energy to maintain homeostasis and you don't need to use that energy, then maybe that capability will get selected out. So here's a tapeworm in somebody's gut. Yeah, that's a human, not sure which tapeworm that is, but that's a human gut revealed by the wonders of endoscopic cameras. Tapeworms don't have any guts of their own, simply because they are bathed in pre-digested food several times a day, and they're capable of absorbing everything they need right through their skin. So we see the complete loss of guts. A lot of parasitic taxa might lose heads, might lose sense organs. Tapeworms don't have eyes or anything like that. We've actually mentioned flukes that on the biochemical level have lost the ability to make some of their bases for DNA because they're able to get everything from their host. You can find some interesting exceptions to the rule. Those are human macrophages, white blood cells, and inside them are a whole bunch of little parasites in the genus Leishmania. Now, I mentioned Leishmania Wednesday in the context of talking about reservoir hosts. Leishmania can cause very severe human symptoms, very severe disease. The species in the Middle East and Central Asia causes a disease called Kala Azar. The South American species can cause severe skin ulceration. And Leishmania, when it's injected into the body by a biting sandfly, Leishmania gets engulfed by the white blood cells, and it's able to live inside the digestive vacuoles of those white blood cells. A part of the digestion process is that inside the vacuoles gets really acidic, and Leishmania can maintain a pH near pH 7 while living inside a vacuole with a pH of 4. So some parasites actually can do a great deal to maintain their own homeostasis without relying on the host to do it. So you do get some exceptions to that rule. But here's the problem, or 3 problems. If you are a parasite, you are living in an island paradise, right, where all you have to think about is lying there and reproducing as much as you can, and your host is playing the role of the very deferential waiter who's constantly coming around bringing you fruity rum drinks. The problems are, 1, how can you be sure you're in the right host? You would not want to walk into the wrong island resort and get kicked out. So how do you know, if you're a parasite, that you're in the correct host? How do you deal with the host's immune defenses? Because, your island resort does have bouncers that are trying to keep out the riffraff. So how do you get past them? And then the really big one is, how do you get your offspring from one island paradise to another? It's great living on that island paradise, but the next one might be 50 miles away and you haven't got a boat. How are your kids going to get across that huge gap? How are, not that this would really happen, I'm just saying here, how would my tapeworm manage to get its offspring into you when you and I are separated by anywhere from several feet to several miles, right? We've got several miles of habitat that is very bad for tapeworms. How do they get from one person to another? The first problem is really freaking complicated, and the details are going to vary a lot between parasite taxa, and in many cases we don't fully understand how they do it. But parasites are capable of recognizing hosts, of knowing, if you will, that they're in the right species, and in some cases of being able to navigate within the host to the right part of the host where they have to be. One thing, I've mentioned this before and we'll see it again, is there are cases when humans get infected by a parasite that is not normally a parasite of humans, and the larvae will sometimes get confused and start wandering through various bodily organs. I mentioned this with the raccoon parasite, where if you swallow eggs, the eggs hatch into larvae, and the larvae will crawl through various organs where you don't want worms crawling, like your brain. Right? So presumably within the raccoon host, those worms have some kind of cues that they can use to guide them where to crawl. I'm not sure where they end up, but they probably have some sort of cue. As far as I know, we're not entirely sure what that cue is. But parasites can use chemosenses, they can use mechanical cues, and they can use behavioral cues to track hosts. I found a nifty example of a chemosensory cue. This is a fish louse. Specifically, it's a salmon louse. It belongs to a group of crustaceans called the copepods. Copepods there's lots of free-living copepods. They're very common in freshwater and the oceans, but there's a lot of parasitic ones as well. And this particular one lives on salmon. There's been a pretty good amount of research done on salmon parasites because farming salmon is a billion-dollar industry now. Most of the salmon that you would most of the salmon I see at the supermarket, certainly all of the cheap salmon that I see at the supermarket, is farmed these days. And there are some concerns about whether that's ecologically sustainable. That's beyond the scope of the course. But when you crowd a whole bunch of salmon into a giant salmon pen, you enhance parasites' chances of transmission, right? For the same reason that when you crowd a bunch of toddlers into a daycare center, everybody gets sick, right? The more hosts you have jammed into one area, the greater the odds of pathogen or parasite transmission. And so a lot of work has been done pretty recently on parasites of farmed salmon. Salmon lice can find their hosts. One way they can do it is behaviorally. They swim towards the light. That's called being positively phototactic. And salmon prefer to stay pretty close to the surface. So if you're a fish louse and you swim towards the darkness, you probably will never find the salmon. If you swim toward the light, you probably will, and then you'll have lots of babies who will inherit your ability to swim towards the light. So here we've got a behavioral cue that's presumably been selected for. But there are chemical cues as well, and this was a paper that came out 3 years ago. A couple of researchers found specific receptor proteins that were expressed in the copepod's first antennae, right? We know they're expressed in the main sense organs that these copepods have. And they figured out that these receptor proteins were involved in identifying hosts because they were able to knock these proteins out using a technology called RNA interference. I think there's more than one way that that can be done, but you can create RNAs that will block the translation of other RNAs, and that's what they did. They essentially knocked out the gene. And when they used RNA interference to knock out a receptor called L-Sal IR25, the knockout salmon lice were more likely to attach to a different species. They were more likely to attach to lumpfish. This is the lower bar graph is the number of lice that attach to lumpfish. You can see that 30 minutes after infection, some of these lumpfish had an average of about, maybe 70, 80, over 100 per lumpfish, but they didn't stay on. They left within 3 days. If you look at the top, if you look at Atlantic salmon, you get lots of lice coming on the fish, and they'd stay there. They'd still be there 3 days later. So when you put a bunch of these copepods in a tank with lumpfish, they'll latch onto the lumpfish, but they leave, except if they have that L-Sal IR25 receptor knocked out, which made them a lot more likely to stay. So if you don't have this receptor, you may stick around on lumpfish, which is not your normal host. So that receptor is clearly involved in figuring out which is the right host. It's the way that these copepods can taste the host that they're on. They knocked out a related receptor called IR8B, and when they knocked that down, that's at this top bar graph over on the far right. The parasites didn't start attaching to lumpfish, but they were less likely to attach to salmon. So knock that one out, and the parasites are less able to attach to anything. Knock out that first one, IR25, and the parasites are less likely to reject lumpfish. So we've got at least 2 proteins, and I'm sure there's more, that are used in chemical recognition that are involved in, fish lice selecting what host they're going to attach to. So there are some cases where we have been able to decode some of that host-parasite recognitions on the chemical and genetic levels. But not all parasites are this good, and this is something that might conceivably affect you if you ever go swimming in swimming holes and lakes and things, which I guess some people still do. There are lots of parasitic flatworms in a group called the flukes that live in snails as an intermediate host. The definitive host will be usually in a vertebrate. The vertebrate will shed eggs from these flukes. The eggs hatch into larvae that penetrate into snails. There will be several rounds of asexual reproduction in the snail. The details are different with different species. We'll get to that later. And what comes bursting out of the snails are many, many thousands of swimming stages called cercaria. There are lots of different looks that cercaria can have. This particular one belongs to a group called the strigiids. We'll talk about them later. The main body of the worm is this head up at the upper left, and then that forked tail coming down. The strigiid will thrash that tail around and try to burrow into any vertebrate that it comes across. There are some species that normally would infect things like ducks. You know, the cercaria will swim around and try to burrow into any duck foot that they can find. But these species will also try to burrow into humans. And they can't permanently infect humans. Your immune system is able to clean them up, but they'll cause a skin irritation called cercarial dermatitis, otherwise known as swimmer's itch. So if you ever go swimming in Grandpa's old swimming hole, or I don't know if anybody would willingly swim in Lake Conway, but maybe if you fall out of your bass boat or something, or if you're my across the street neighbor, your crappie boat, because that's what he fishes for, or something like that. If it happens at the wrong time of year, you can end up with a fairly minor but rather annoying skin rash for a couple of days caused by cercaria trying to burrow into you. They don't succeed, but they cause irritation until your body can clean them up. So evidently these parasites aren't that good at host detection. Either that or we taste like ducks. Okay, the other problem is you're on this island paradise, but how do you get your offspring to the next paradise, which is isolated and enormous distance from you? To increase your odds, parasites are noted for producing utterly insane numbers of offspring. If you produce just one egg, it may be very unlikely that that one egg will ever make it to a new host. So you increase your chances by producing millions upon millions upon millions of eggs. And the quickest way to do that is through asexual reproduction. Now, a fellow named John Maynard Smith, one of the founding fathers of the modern synthesis of evolution, pointed out a seeming paradox. Sexual reproduction is extremely common throughout life, and yet it puts you at a disadvantage because it takes longer. You have to find a mate. There may be costs associated with that, and it takes two individuals to reproduce. If you're asexual, it takes one individual to reproduce. So if you had a population of mixed sexuals and asexuals, the asexuals should be able to reproduce twice as fast because they can bud offspring off, whereas the sexuals actually have to pair up and it takes 2 sexuals to make a baby, whereas it only takes 1 asexual to make a baby. We actually see this in parasites because many of them will go through multiple rounds of asexual reproduction, which creates potential for enormous exponential growth. You'll see examples of this all through the course, where a lot of parasites have a stage in their life cycle where they'll reproduce asexually and sometimes do it several times. So one parasite might produce 100 offspring, each of which produces another 100, each of which produces another 100, and so on. We talked about those cercaria just a minute ago. A snail might get infected by only 1 larva, and it may shed hundreds of thousands of cercaria because there'll be 2 or more rounds of asexual reproduction. But sexual reproduction in parasites also has to be ramped up. And a lot of parasites will switch it between an asexual stage and a sexual stage. In fact, we talked about this earlier when I was talking about definitive versus intermediate hosts, right? The definitive host is where the parasites reproduce sexually, and the intermediate host is where they reproduce asexually. And many successful parasites crank out the babies like it's going out of style in both. Lots of sexual reproduction, lots of asexual reproduction. That tapeworm I showed you earlier doesn't have much in the way of sense organs, doesn't have a gut. It's basically one giant chain of gonads, and that's about all it does. And many that switch between asexual and sexual stages turns out to be common in species that exploit very rich resources that are also very widely scattered. These are aphids. These are insects that suck plant sap. And aphids are noted for reproducing parthenogenetically, which just means that females produce eggs that are fertile without the need for male intervention. Aphids can also have really short generation times. I believe there are some aphids where the eggs are incubated in mother's body, and the baby aphids are starting to produce offspring while they're still inside mom. You can have 3 generations nested inside each other like Russian dolls. There's also some mites that do that sort of thing. It's really pretty horrifying. There's also a group of mushroom midges. Midges being little flies like no-see-ums and things like that, that do the same thing. They have a stage where the larvae become, the ovaries develop extremely fast and the larvae become pregnant while they're still larvae, which is pretty awful. And the point of all of this is that aphids and mites in the genus Pyomotus and gall midges in the genus Mycophylla are taking advantage of a resource that's incredibly rich but isn't there for a long time. Could be plant leaves. In the case of those mushroom midges, it's mushrooms, right, which sprout up very fast, right? If you've ever gone out on your lawn and seen a whole little forest of mushrooms come springing up, but they don't last very long. So critters like this will reproduce asexually when there's a rich resource available because it's adaptive to do it, right? If they don't take advantage of that resource, somebody else will. So selection favors individuals that can crank out the babies as fast as possible, use up that resource, and then switch to sexual reproduction when the resource starts getting scarce. And sexual reproduction in aphids and gall midges produces winged adults that can disperse and go on to the next plant or the next mushroom, whatever it might be. So in parasites, it's the same thing. If the parasite's to have any hope of reaching a new host, it may have to reproduce asexually in order to take maximum advantage of that host, and it may have to reproduce sexually an awful lot in order to get its eggs over to a new host. This is a tapeworm parasite of sperm whales called Tetragonoporus calyptocephalus. For obvious reasons, we don't know an awful lot about it, right? You don't want to go, killing a bunch of sperm whales just to see what's in their guts. So these are not often seen. This is a picture of a juvenile. Up at the top, that's an attachment structure to attach to the wall of the gut. And this juvenile is only a few millimeters long. When this is an adult, it can reach 130 feet. Lots of room in a sperm whale gut. And tapeworms are divided into segments called proglottids, which are packed full of gonads. And if you're a tapeworm, you're constantly creating new proglottids towards your head end. And as they mature, they pass towards the tail end and eventually break off. So in some cases, the proglottids may break open and release their eggs into the feces. In other cases, the proglottids may come out whole. If you've ever, as I have, had the dubious pleasure of taking care of a dog that's not been very well cared for, and you've ever had occasion to see that dog poop out something that's kind of whitish and wriggles a bit. You don't have to tell me if that's happened to you, but it does happen. That's actually a chain of proglottids, and the proglottids are packed with gonads, and by the time they're excreted, they're packed full of eggs. So every proglottid of Tetragonoporus contains 14 gonads. Each one of the ovaries might contain, I don't even know, thousands of eggs. An adult Tetragonoporus may contain 45,000 segments, and segments are constantly breaking off the tail end of the worm as new ones are added at the head end of the worm. So I'm not even sure I can do the math, but this thing must be producing millions upon millions of eggs, I don't know, every week, maybe every month. And I have no idea how long this thing can live, but you can see that the reproductive potential is absolutely tremendous. As someone pointed out, there are few whales and the ocean is large. I don't think we know the full life cycle. But, if you're living in a sperm whale and you want to get your eggs to another sperm whale, other sperm whales might be very far distant and the ocean is a huge place, so you've got to crank out as many eggs as possible. I found this out. There is the common human intestinal roundworm, Ascaris lumbricoides. In 1 year, a mature female Ascaris lumbricoides, they're fairly big. They can get up to maybe about 10 centimeters long. They're big enough that if you have lots of them in your guts, they can actually cause blockage, and that's that will kill you unless you can get quick surgical intervention to get that blockage removed. Each one can produce 65 million eggs a year. The weight of 65 million eggs is 1,500 times the weight of the actual worm. So that gives you some idea of how much reproductive potential they've got. Imagine being able to reproduce. This is really gross if you think about it in people, but imagine being able to reproduce 1,500 times your own body weight worth of babies. Like, dang. But again, that's what you have to do if you're a parasite. I mentioned earlier that twofold cost of sex worked out by John Maynard Smith. And he showed that asexual reproduction can ramp up faster because in an asexual population, everybody can reproduce. Everybody can bud off a new offspring or lay pre-fertilized eggs or whatever it might be. While in a sexual population, individuals have to pair up, so an asexual population will grow twice as fast. And Maynard Smith asked the question, why is sex evolutionarily favored? What's so great about sex? Insert punchline here. We pointed out that sexual reproduction is nearly universal. Certainly it is in the animal kingdom. There are some animals that reproduce entirely asexually, but there's not many of them. There's one group of about 300 species called adeloid rotifers. If you take me for invertebrate zoology, you'll get to meet these guys. There's a few, there's a couple of others, but it's not common at all. There are lots of species that can switch hit, like those aphids we were talking about earlier, but there's very few that exclusively reproduce asexually. Smith's answer as to why sex is so universal, even when an asexual population can outbreed a sexual population, is that sex must be, evolutionarily speaking, really good for you. And it's good for you because it generates variation. Like these kittens. Sorry, after talking about parasitic worms, I thought we might appreciate some kittens for a moment. Next week, we'll talk about the horrible things that live inside these kittens' intestines, but for now, just enjoy the cute. Sex generates genetic variation. Sex, stripped of everything else, is two individuals doing genetic engineering with their DNA. Combining DNA from both of them gives offspring that have new combinations of alleles, right? New phenotypes, new traits, all of that. It allows organisms to repair their DNA. If I've got a mutation on one of my chromosomes, there is at least a chance that I can reproduce with somebody who doesn't have that mutation, and my offspring won't have it either. One of the problems, I don't know how much time I have to spend on this, but one of the problems with asexual reproduction is something called Mueller's ratchet, which is asexual reproduction is not efficient at repairing DNA damage. You know, for the same reason that if you were to make a photocopy of a page and then make a copy of a copy and then a copy of that copy of that copy and so on, after a few generations, the page would be completely illegible because at every step, you introduce noise, right? You introduce flecks and distortions and things like that, and there's no way of getting rid of them. So if you're reproducing only asexually, you tend to accumulate DNA damage, and you can't get rid of it. In sexual reproduction, you can, because you're basically blending your DNA with somebody else's to create offspring that at least have a chance of not inheriting your DNA damage. So sex makes you variable, and sexual reproduction allows organisms to get rid of negative mutations. Now, a guy named Lee Van Valen, not to be confused with Van Halen, by the way. I don't know if he's a good guitar player, but I don't think he's noted for his two-handed finger tapping technique, as you heard on Eruption. Lee Van Valen came up with what he called the Red Queen hypothesis. If you're wondering what the Red Queen is, you might have known Alice in Wonderland. That was a book by Lewis Carroll, Alice's Adventures in Wonderland. And you know this if you've just seen the movie, but in Alice's Adventures in Wonderland, she gets caught up in this weird card game. Right? Remember, there's the queen, there's the duchess, there's this pack of cards that are, you know, running around, bending themselves into croquet hoops and all of that. I assume you've heard of it. There's a follow-up called Through the Looking Glass, where Alice gets trapped in a weird world that's basically a giant chess game. And she encounters the Red Queen, seen here on the right. Chess sets at the time were white pieces versus red, rather than white versus black. So I'm not sure why that color changed, but yeah, white and red are the sides here. And Alice is a pawn, so she's trying to get made a queen herself and get to the back rank. And the Red Queen suddenly yells, "Hey, we have to run." And they start running as fast as they can. And then Alice looks around and nothing has changed. And Alice says, "You know where I'm from, if I run, I usually get somewhere." And the queen says, "That's silly. Here, you see, it takes all the running you can do to keep in the same place." That's why Van Valen calls this the Red Queen hypothesis. Think about it. You're an organism. You've got a niche that you live in. There are other organisms that would love to take that niche from you, so you've got competition. There are organisms that would love to take your resources, so you've got predators, you've got parasites, you've got pathogens, bacteria and things like that. All of these organisms that are trying to take what you've got. And all of those organisms are subject to evolution by natural selection. They're constantly evolving. So at any given moment, you might get a predator that's evolved the ability to penetrate your armor, or a pathogen that's evolved the ability to make you sick, which it didn't used to be able to do. Exhibit A, COVID-19. Or you might get a parasite that has evolved the ability to get past your defenses. Or you might just get a competitor that has evolved a slight advantage. So his point is that you have to evolve, right? You're under constant selective pressure. New alleles, new gene combinations constantly have to be made just for you to keep the position that you've got, just for you to be able to keep occupying your niches, right? So if a predator evolves teeth that can break through your skin, that puts you under selective pressure to evolve thicker skin, which puts selection on the predator to evolve sharper teeth, which puts pressure on you to evolve thicker skin, and so on. You get what we call an evolutionary arms race. What this has to do with sex is that sex generates variation, it generates new combinations of alleles, and new combinations of alleles, all that variability is exactly what you need in a world where everybody is trying to take where you've got, what you've got, and it takes all the running you can do to keep in the same place. This has been experimentally shown several times. These are water fleas in genus Daphnia, very common in fresh water around here. We use them in 1400. I'm not sure if we do in 1440 or 1441, but we've actually got a colony of them in the greenhouse, I believe. And on the left is a healthy Daphnia. On the right is a Daphnia that's looking cloudy because it was infected with bacteria in genus Pasteurella. The one on the right is a very sick Daphnia. Daphnia can switch between producing offspring sexually and asexually. Sexually produced offspring turned out to be over twice as resistant to bacterial infection as asexually produced offspring. Think of it this way. Let's say we give you all computers, and all of them have the same password. Password 123 or something like that. If a hacker cracks one of your computers, he immediately gets access to all of them because they've all got exactly the same password. On the other hand, let's say that everybody's got a different password and you're constantly changing your passwords once every month or something like that. Then if the hacker cracks one, he can't crack all of them, and he may make some progress on cracking your password, but he may not have time to crack it before you've changed it. So sexual reproduction is like a world where everybody is changing their passwords all the time. Hosts are constantly coming up with new combinations of alleles. They're varying a lot, and that's a moving target for a parasite or a bacterium to hit if the host is constantly scrambling its DNA by mixing it with somebody else's, which is what sex basically is. It makes life much more difficult for parasites and pathogens. Pretty well-known study was done back in the 90s of a freshwater snail in New Zealand called Potamopygus antipodarum. This is another species where you can have sexuals and asexuals coexisting. And there's a lot of scatter in the data, but on the y-axis in this graph, you're seeing the frequency of males. So you can see some populations are all female, and some are getting closer to 50-50. And there's some scatter, but this is statistically significant. Sexual individuals are most common in populations that have a high parasite load. That's on the x-axis. That's the incidence of parasite infection. So that supports the hypothesis that sex is an anti-parasite defense. On the left, by the way, that's a map of New Zealand, and those things that look like pins stuck into it, the head of each pin is a little pie chart. Probably hard to see, but it just shows the incidence of sexuals in each population. That little white spot is the percentage of males in the population, and each one of those little pinheads is a tiny little pie chart. It's not easy to see, but you can read the graph pretty well. Oh, incidentally, this matters because when snails get infected, what often happens is that the parasites will absorb so much energy that they end up devouring the snail's own gonads. It's called parasitic castration. And I said parasites don't kill their hosts, but if they eat your gonads, you're not going to be doing any reproducing. So a snail that gets heavily infected with these flukes may live, but it will never pass on its genes. So there is high selective pressure in favor of parasite resistance. And one way to do that is to reproduce sexually. That's exactly what we see. Incidentally, this New Zealand snail has turned up in the Pacific Northwest, and it seems to have left behind its parasites. Populations in the Pacific Northwest don't have any parasites on them. We predict that asexuality would be favored because if there are no parasites, then asexual individuals have that advantage because they can make twice as many babies as the sexuals can. Somebody ought to look into that. I don't know if we actually see that, but it would certainly be interesting to find out. Okay, and again, parasites often reproduce asexually because they have rich resources, but probability of finding, sending offspring to new hosts is low. So they'll use those rich resources to crank out lots of babies. This is Plasmodium, the causative agent of malaria. Plasmodium gets injected into your blood by a mosquito bite, penetrates into cells in your blood, also in your liver, think it starts by going through your liver, but it'll go through rounds in your blood cells as well, live inside that cell, and then divide into a bunch of tiny swarm cells called schizons that go bursting out of your blood and liver cells and go on to infect new cells. And malaria can do this for years. But because parasites can't adapt to changing host defenses if they reproduce only asexually, Plasmodium goes through a sexual stage that happens in the body of the definitive host, the mosquito. We'll talk later about the guy who discovered this and worked out the life cycle and won the Nobel Prize for it and wrote poetry about it and was a real jerk to his competitors. But that will come later. We'll talk about Sir Ronald Ross. Last thing, brief illustration that sexuality is adapted for the parasite as well as for the host, right? If your host is constantly changing its genetic password, then the parasite has to constantly change its strategies as well. There's a cat eating a mouse. Mice can be infected with a protist called toxoplasma. It's actually related to Gregorines and to the malaria parasites. Humans can get this as well, by the way. And some German researchers found a cat that had been infected with 2 different strains of toxoplasma at the same time. And they found that the 2 strains were actually hybridizing within the cat, right? The 2 strains were hybridizing within the body of the cat. They tested the resulting cysts for infectivity. What you're looking at is each one of these collections of bars corresponds to a different genotype that was produced by sexual reproduction between these 2 different strains. And they varied tremendously in their ability to infect the intermediate host, which is mice. Some of the strains of toxoplasma that were produced by the sexual reproduction, the ones on the right were not very good at it at all. The ones on the left were extremely good at it. You can see those bars are up there a lot higher. And so sexual reproduction of the parasite created variation within the parasite. And some of those variants were not very good at infecting mice. Some of them were extremely good at infecting mice. So sexual reproduction is important for hosts to defend against parasites, and it's important for parasites because that's the way they can develop new abilities to infect hosts. So sex is basically an arms race. Sorry, I'm having a Pat Benatar moment. Love is a battlefield. Ah, love is a battlefield. Okay, sorry about that. Every so often it just comes over me. Okay, right, I'm going to go ahead and stop, collaborate, and listen. Sorry, stop recording. And I'll see you folks on